microscope, before genetics, before we even understood reproduction, people still try to explain how life begins. Where does a human came from? And how does something so complex start so small? So different ideas emerged. In ancient Greece, thinkers like Aristotle supported epigenesis, the idea that an organism develops gradually, step by step, forming structures over time. Centuries later, another belief appeared, preformationism. This idea claimed that a tiny, fully formed human, sometimes called a homunculus, already existed inside the sperm or egg, just waiting to grow. At the same time, many believe in spontaneous generation, the idea that life could arise from non-living matter. Different theories, different explanations, all trying to answer one question. How does life begin? Spontaneous generation was a belief that life could appear from non-living things. Rotting meat produces maggots. Dirty water produces tiny organisms. It seems logical. For nearly 2,000 years, people believed it, until Francisco Redi tested it. He placed meat in 2 jars, one open, one sealed. Maggots appeared only in the open jar. Later, Louis Pasteur proved that microorganisms come from other microorganisms, not from nothing. And just like that, spontaneous generation was replaced by biogenesis. Life comes from life.
Imagine if this entire landscape was never actually built. No planting, no shaping, no gradual growth. Imagine it was always complete, just compressed, hidden, waiting to expand. That was the logic behind preformationism. In the 17th and 18th centuries, scientists such as Marcello Malpighi supported this view after observing chick embryos. Some, like Nicholas Hartzucker, even drew images of a tiny human inside a sperm cell. They believed the embryo was already complete in miniature form. Development was only growth in size. No new organs formed. No new structures appeared. Everything was pre-made from the beginning. It was a neat explanation, but it struggled to explain how new features actually arise during development. Look at this field. It did not begin as a perfect course. It began with seeds, bare soil, incremental change. This reflects epigenesis. In ancient Greece, Aristotle proposed that organisms develop gradually. Structures form step by step, cells organize, tissues emerge. Organs take shape over time. Later, in the 18th century, Caspar Friedrich Wolff provided strong evidence for epigenesis by also studying chick embryos, developing a theory opposing Malpighi. He observed that organs were not preformed, they developed progressively. Epigenesis argued that development is construction, not enlargement. Once microscopes improved and experiments became more controlled, science began shifting from assumption to observation. That shift would lead directly into the birth of scientific embryology.
For thousands of years, humans debated life of how it begins without actually seeing the thing that starts life, which is like reviewing a movie that you've never watched before.
In 1655, Robert Hooke made a groundbreaking observation. He examined cork under a microscope and noticed tiny box-like structures. He dubbed them as cells, not for their scientific allure, but because they resembled the small rooms of a monastery or a prison. Ironically, these cells would later confine countless generations of biology students in the lecture halls and even textbooks.
Robert Hooke had unknowingly introduced the structural unit of life, but he did not discover reproduction. Instead, he simply made the life become what we call visible. And then came Anton van Leeuwenhoek, a man who built a better microscope, wherein he looked at what we called the sperm cells.
He described them as tiny, moving animal cues, which sounds friendly. They were not actually friendly. They were evidence for the first time in history reproduction was no longer philosophical. It was cellular. Life was not forming from abstract forces. It involved actual physical units, that is, moving, dividing, and even organizing.
But observing cells wasn't the only beginning. Because once the scientists see something, they inevitably ask, what if we interfere? Once the microscope revealed that the embryo came from the cell, scientists had a new idea, which is to test them.
In the late 1800s, William Rox conducted a pivotal experiment on a frog embryo. He deliberately destroyed one of its two cells carefully. The outcome? Only half of the embryo became developed, and from this, Rox concluded that each cell had already predetermined fate, and this phenomenon became known as what we call the mosaic development. Cells were essentially pre-programmed. But biology rarely cooperates with simplicity. And then enter Hans Driesch, a German embryologist in the late 19th century. He separated a sea urchin embryo at the two-cell stage. Instead of forming half an organism, each cell developed into a complete, though smaller, embryo, directly challenging Rox's mosaic theory. Driesch called this as a regulative development. It demonstrates that early embryonic cells are totipotent. Each can generate a cell all types of organism rather than being locked into a fixed rules. And these cells are flexible and capable of reorganizing after a disturbance. In short, early embryos can compensate, adapt, and still construct a functional organism that is essentially the overachievers of developmental biology.
By the end of the 19th century, humanity had learned two critical truths. The first one is that life begins in the cell. The second one is that the cell can be influenced. And then we begin to realize that the cell can be influenced. We can then wonder, can it be directed? Can it be reprogrammed? And lastly, can it be re-engineered?
Hi, this is a laboratory. This is where scientists try to understand life, which is quite ambitious considering that most of us are still trying to understand group work. But here's a serious question. If all of the cells in your body have the same DNA, why isn't your brain shaped like your liver? Or why doesn't your eye randomly grow on your elbow? And how can one fertilized egg know how to build all of this? Developmental biology tries to answer that. And surprisingly, the answer isn't magic, it's control.
At the center of development is DNA, not just as a blueprint, but as a carefully managed instruction system. Genes don't just sit there. They're turned on, turned off, activated at specific times. During development, certain genes tell cells, you are going to be muscle. Others say, you form the spine. Specialized genes like Hox genes determine where structures appear in the body. Development isn't random growth. It is a coordinated genetic performance. Like an orchestra, except instead of violins, we have transcription factors. But here's where it gets interesting. If genes can be switched on and off, can we switch them ourselves?
Enter stem cells. These are cells with options.
Unlike most adult cells that are permanently specialized, stem cells can become many different cell types. In early embryos, stem cells are pluripotent, meaning they can form nearly every tissue in the body. Even more astonishing, scientists discovered that adult cells can be reprogrammed back into this flexible state. It's like convincing a fully trained engineer to go back to kindergarten and start over. This discovery changed medicine. If we can guide stem cells properly, we might repair damaged tissues, replace lost neurons, and restore heart muscles after injury. But if cells can be reprogrammed, could an entire organism be recreated?
In 1996, scientists introduced the world to Dolly the sheep. She wasn't created through traditional reproduction. She was cloned, created using somatic cell nuclear transfer.
The nucleus of an adult cell was inserted into an egg cell, and remarkably, it developed into a complete organism. This proves something extraordinary. Even a fully specialized cell still contains the genetic blueprint. Developmental potential isn't erased, it's regulated. Cloning forced humanity to confront new questions. Just because we can create life, should we? More importantly, can this be knowledge be used for healing rather than replication?
Today, developmental biology has entered the era of regeneration. Scientists are trying to find how tissues repair themselves. Some animals can generate their limbs, humans cannot yet.
Through tissue engineering and stem cell therapy and 3D bioprinting, researchers are attempting to grow functional tissues in laboratories. The goal is not just to understand development, it is to control it safely, precisely, and ethically. From repairing spinal cord injuries to growing replacement organs, regenerative medicine represents the future of developmental biology.
Modern developmental biology isn't just about how life begins. It's about how life is directed, controlled, reprogrammed, and possibly repaired. What started as ancient debates about tiny humans inside cells has become a field capable of editing genes, reprogramming cells, and regenerating tissues. And maybe the most fascinating part. Every answer we discover creates even bigger questions, not just about how life develops, but about how far we are willing to shape it. And that might be the most human experiment